Any kind of drug has a difficult time penetrating the blood-brain barrier. So what does that mean for patients, and how are you getting around that obstacle with the types of therapies and the radiation-directed therapies you're developing?
2% of drugs are thought to cross the blood-brain barrier. We're getting 100 to 1. So we get almost zero systemic delivery. So we can give the maximum of 740 grays of radiation in a single dose to the brain without any untoward cranial, intracranial issues.
A test where we take an LP, lumbar puncture, so we take a CSF, cerebral spinal fluid specimen. We process that in our centralized lab here in Houston, and we can detect cells, in a case where other technologies don't detect these carcinoma cells, we can get them down to, 1 or 2 cells in a whole draw from cerebral spinal fluid, which is, exquisitely sensitive.
How are you getting around that obstacle with the types of therapies and the radiation-directed therapies you're developing?
We're getting 100 to 1. So we get almost zero systemic delivery with 20x the kind of radiation you can give to glioblastoma versus EBRT with almost no systemic toxicity. And that's really because we don't have to push the drug through the blood-brain barrier.
So Marc, Russell, welcome to the podcast today. Really excited to meet you. And I thought maybe before we get started here, maybe each of you could give a brief intro into your role and what you do.
Yeah. Hey, Chris. Thanks for having us on. So my name is Marc Hedrick. I'm the president, CEO of Plus Therapeutics. My background started as a physician. I was an academic surgeon at UCLA, doing about half research, half clinical work. And part of our research was in the stem cell therapy area, which was kind of pre-2000, and had the opportunity to take a 1-year sabbatical, which has now turned out to be about a 26-year sabbatical. So I suspect my office has been given away by now, but been doing this, love it. We've never been happier doing what I'm doing, and we're so close to doing something really special. So we're excited to talk about it.
Thanks, Marc. And how about you, Russ?
Yeah, Chris, thanks for having us on. It's good to be here. Russell Bradley is my name. I'm the president and general manager of the CNSide part of Plus Therapeutics business. Yeah, we're focused on the diagnostics, and Plus is a therapeutic company. Yeah, my background is a diagnostic and life science executive. Pretty much my whole career outside of college, I was a science grad back in the day. Worked at Abbott Labs for a while in their diagnostic division. Learned about Plus and what they were trying to do with this test. Became convinced that this was something we had to get to market for these patients, and it's a privilege to be able to work here every day and get that done for them.
Terrific. And where are you both based? I mean, now, Marc, you mentioned you UCLA. So you were in California for a while, and where are you working now?
Yeah, I've come full circle. It's not quite the 40 years in the wilderness, but I left Texas after medical school and basically went to California for 30 years with a few training stints at different places, but that was about 38 years ago. So I'm back in Houston and relocated here, actually. So
Okay. Awesome.
Yeah, that's where the company's based currently.
Your company's based in Houston. So yeah. Russ, are you in the Houston area as well, or are you working elsewhere?
Yeah, I'm based here, Chris, in Houston. I'm here in the lab today. It's a great place to locate a lab. We're less than a mile from the Texas Medical Center, which is the biggest medical center on the planet. Originally from Australia many, many years ago. Lived in California twice, Florida for a while, Texas twice, and happy to be here.
Yeah, terrific. So I think, Marc, maybe I thought I'd ask you to give an intro into the company, the mission, and also how you're structured. I know we have Russ here on the diagnostic side. You've also got a therapeutics portfolio. So maybe just give us an idea of the mission and how you're structured before we jump in.
Yeah, we're unique, and I appreciate the opportunity to be able to explain this a little bit more. I think it makes complete sense once you sort of understand the context, but Plus in its prior version was one of the first public stem cell companies in the US. And my mandate with the board when I took this role was to make that work, capitalize it and grow it, or divest it. And it became very difficult. Even though the data was positive, it became difficult, in my view, to fully capitalize it like it needed to be capitalized. So we divested that technology. We had some oncology technology And around that time, the board chair and I really felt like there was an opportunity to build off of one key concept, and that is that central nervous system cancers are known to be largely a death sentence to patients. However, the best killer of brain cancer is radiation, but the amount of radiation you can give is highly constrained. And so we felt like if we could find a way to increase the amount of radiation to the CNS cancers, both metastatic and primary, that we can make a material improvement in survival. And that led to a couple of acquisitions. And thus far, we've done about 5 licensing agreements or acquisitions. And so what we've built now is a comprehensive CNS healthcare company with a diagnostic group, with a therapeutic group, and then silently but steadily, we're working on an advanced data analytics behind the curtain, because we recognize that by virtue of mostly the diagnostic, but also therapeutic, we're going to have a tremendous amount of data on CNS cancers that nobody's ever collected before. And I think our ability to continue to drive the pipeline with that data is enormous. So there'll be sort of 3 legs of the stool, and Russ runs the diagnostic component.
Yeah, Russ, do you want to jump in and provide a quick overview from your end as well?
Yeah, so Plus is a very unique company to have diagnostics and therapeutics under the same roof. It makes sense when you think about it conceptually, but it's just so rare. Usually it's the preserve of huge companies like Hoffman and Roche, the very largest companies, that to be able to really run both of those together. But then they have the challenge of trying to focus it, because it's, oncology itself, therapeutically and diagnostically is a hard thing to tackle. What we've got is a very focused linear, and we can focus on a particular indication and patient type, which allows us to bring critical mass. So the part that we have here is really part of a virtuous circle, if you like. We've got a very sensitive diagnostic in a marketplace where the standard of care is just inadequate. And it's not my view, that's the view of the physicians that are servicing and supporting these patients. So we've got this very sensitive diagnostic that allows us to find very rare cell events in the cerebral spinal fluid of these patients. And that allows us to get them on treatments as we characterize those cells sooner and impact outcomes, on longer term outcomes. And so the combination of a company that's got a focused therapeutic on this patient population and a diagnostic that's unique and powerful, and plus, as Marc said, the data analytics, you bring it all together. And we think we can bring a lot of power to bear on this focused population and really impact the outcomes.
Terrific. I think to get started here, I think what we'll do is let's focus a little bit first on the therapeutic side, and before we dive into the pipeline, Marc, I think let's talk a little bit about exactly the problem. For the listeners out there who may not be so familiar, we know that many drugs, cancer drugs, and any kind of drug has a difficult time penetrating the blood-brain barrier. So what does that mean for patients, and how are you getting around that obstacle with the types of therapies and the radiation-directed therapies you're developing?
Yeah, let me start that answer with kind of acknowledgement of the unique biology in the central nervous system. You mentioned the blood-brain barrier. The immune system's different. The anatomy is kind of funky with the gyrations. You've got the brain, but you've got all these kind of very complex subsections and neural connections. It's encased in this very hard piece of tissue, and then you've got the skin, and that's one reason we think that pulling it all together under one roof is synergistic. So, but I acknowledge the blood-brain barrier is a challenge. There are ways around it, but let me start then with sort of the therapeutic approach. So I think it's helpful to understand the market first, and the market and epidemiology. I'm sort of using those as substitutionary terms, but there's an epidemic of CNS cancers. Most people aren't aware of that. You don't see it on the news, but you talk to neuro-oncologists, they don't not all of them say it that way, but that's what they're saying. And there's been an explosion, largely in metastatic disease, because there are better treatments for common cancers like breast cancer, lung cancer, mostly carcinomas, melanoma, and so forth. So these patients are living longer, and the end game to their life is becoming CNS oncology related CNS mets. And so if you think about it as a whole, about less 5% of all CNS cancers are primary, the rest are metastatic. And parenchymal brain mets, metastases to the brain tissue itself, have been sort of the leading cause, but leptomeningeal cancer has actually caught up with it and surpassed it, and maybe significantly so because it's underdiagnosed. Hence the reason we acquired the diagnostic. So the real commercial opportunity, in our view, is to take that group of metastatic patients and then try to turn that into the chronic disease that their primary was. I think there's a real opportunity to do that. So let me talk about CNS targeting. So the blood-brain barrier is a problem, as you've sort of alluded to. And 2% of cancers or 2% of drugs are thought to cross the blood-brain barrier. So that's complex biology. People have tried and failed at that, and you can sort of tune it in a way, but it's not great, it's not reproducible. And so many cancers are infiltrative in the brain, and so it becomes a real challenge there to target the blood-brain barrier. There are actually a couple of surgically related ways to bypass the blood-brain barrier, and we felt like to make an impact in the market today, we needed to not do years and years of biologic research, but to take things that are known to work, take the blood-brain barrier out of the game, and allow us to get the punch to the other fighter. And for parenchymal disease, glioblastoma, parenchymal brain mets, there's something called convection enhanced delivery. Let me just flag that, and I'll explain that if you'd like. That allows you to get lots of drug right onto the tumor in a very, very controlled way. And that's a commercially available technology. And with leptomeningeal cancer, which is a cancer of the fluid around the brain, something called an Ommaya reservoir that gives you two-way access to the fluid around the brain, like a chemotherapy port would. So there are ways to tackle the blood-brain barrier that aren't necessarily druggable, but are very effective.
So tell us a little bit then about your lead asset that looks like you're evaluating, RayObic, in a number of different, one or two major indications in the CNS system. So maybe you start there before we talk about the nanoliposome therapy.
Yeah, so RayObic it's a complex three-part formulation. So this starts with a radioisotope called rhenium-186 and has the ideal physics for the central nervous system. If you look at its various physical properties, it's almost ideal. And the chemistry works with how we formulate the drug. We have a proprietary chelator that grabs really any heavy metal, but does really well with rhenium and allows us to load it into lipid constructs. And so the drug is really a radioisotope, chelated, many of them loaded into an nanoliposome, and the purpose of the liposome is to change the pharmacokinetics. So then when we place that drug either by convection enhanced delivery or by Ommaya reservoir into the CSF fluid, essentially all of the radiation goes to the region of interest. So when I talk to other radio farm companies or their leadership or hear their presentations, they talk about their therapeutic index, and they're trying to get 3 to 1, 2 to 1 in some cases. We're getting 100 to 1. So we get almost zero systemic delivery with 20x the kind of radiation you can give to glioblastoma versus EBRT with almost no systemic toxicity. And that's really because we don't have to push the drug through the blood-brain barrier. We parachute beyond it, deliver max amounts of radiation. The brain actually is very radio tolerant. We're giving doses that no one has ever given before deliberately into the brain. And so the brain is very radio tolerant, we found.
I just want to follow up because I know rhenium is sort of not your typical radio ligand that or radioisotope that's used for many therapeutic uses. What kind of made you choose that isotope over others that are commonly used?
That decision predated me to the team that originally developed it at MD Anderson, the University of Texas. But the idea is that for the CNS in particular, you wanted something that had the right path length, and rhenium's got a great path length. Beta particles, so like high-energy electrons, which are the killing component of the rhenium, are very effective in terms of DNA double-stranded breaks. The half-life makes the supply chain, very, very doable. And in other words, from a single site, we can supply the entire US and probably some major European and Asian capitals. The energy level is kind of in sort of a mid-level, and the chemistry I mentioned before, but you have to have the right loading chemistry. Unlike an alpha particle where we don't need to get the radioisotope essentially abutting the cell. We want to get the drug to the region of interest and let it fully decay. It decays fully over 2 to 4 weeks, and that formulation keeps the drug there. So the final property of rhenium is it just matches the formulation. An alpha particle wouldn't work in this construct. It would work in other constructs perhaps better, but the rhenium is ideal for this. And then the handling is very easy. The path length is a couple of millimeters. And there's some biologic reasons why that's the absolute sweet spot for leptomeningeal cancer. So you go down the property and set up your table, and rhenium just wins out, across the board.
Just one extra follow-up question, Mark, on that, because it's interesting. The first approved radioligand therapy suffered from sort of lack of sort of isotope supply and to meet the demand in the market. Like, are you supply constrained? I mean, how are you securing the isotope supply for your clinical trials and then thinking ahead to commercial supply? Is that going to be an issue for rhenium? Because I just don't know. I'm not that familiar with this particular isotope.
The only supply constraint today that we have to manage is just radiation services. So the target is placed in a reactor. We use MUR. We rent space in MUR's reactor currently. We're not commercial. MUR is a commercial reactor. I think they're one of the biggest producers of lutetium. So the key is just having a backup supply to that so that we don't ever go without a product. And we're working on that. So once that's in place, then the whole supply chain will have built-in redundancy, and we'll be able to supply any sort of conceivable scenario once we're commercial that we can envision.
Okay. So Mark, walk us through your current learnings from, I think you have a couple of clinical trials ongoing that respect LM phase 1 study. Just like what can you share with us publicly about the profile of the asset in the clinic?
Let me tell you a couple of things that sort of cut across, all the indications that I think that are scientifically important. And if anyone's interested, I refer them to the Nature Communications paper that we authored year and a half ago, summarizing the phase 1 data for recurrent glioblastoma. And one thing that we learned is and I sort of alluded to it before, the brain is incredibly radiotolerant. So we can give the maximum of 740 grays of radiation in a single dose to the brain without any untoward intracranial issues. You're not getting, the brain is not melting. You're not getting, massive neurotoxicity. The patients are doing very well. They have no decline in their neural function, even at very high levels. So the higher you go, or higher one goes, you don't see an increase in neural-related toxicity. And just to kind of put that in perspective with what is currently done in the clinic, for primary GBM and recurrent GBM, the range of radiation delivered or absorbed is about 35 to 50 gray, fractionated over 6 weeks. So we're giving, a log more radiation in a single shot. The brain's very tolerant to that. That's point number one. The other thing that we learned, I think it's important across all of the indications we're looking at, and I think important for the field, is that we've yet to reach a maximum tolerated dose. We haven't gotten to a safety signal in either of our trials. It goes back to the radiotolerance of the tissue, but perhaps it says something about nuclear radiation versus photon beam therapy. But we can give extremely high doses, and as we escalate dose in our phase 1 trials, we see a highly statistically significant correlation between dose and overall survival, not even with an intermediate endpoint. Overall survival correlates very well with dose. So we haven't reached a max yet. So it says that we've been operating very low in the therapeutic range as it relates to radiation. It sort of to me, it sort of supports our original notion that, can we really turn up the dial in radiation and get better outcomes? And I think that's what we're showing.
So you've completed a phase 1 study, right? So what you're saying is what you've learned so far in the clinic is that it's safe. You haven't reached a maximum tolerated dose. Any efficacy signals yet that you could speak to, or is it still too early?
Yeah, thanks for following up. So I'm just kind of talking about big pictures. Let's talk about kind of more specifics now. So we're on the downhill slope of a phase 2 for recurrent GBM. The phase 1 showed that we doubled survival over standard of care. And a cut of the phase 2 data midway through showed about a 60% improvement in survival. We know that in recurrent GBM, no matter what you do to patients, no matter what clinical trial they're in, you take a big group of patients, the median overall survival is 8 months. That's it. And so we're doing significantly better than that in the phase 1 and midway through the phase 2. So yes, and the endpoint there being overall survival. In LM, in the phase 1, we also showed a trend towards improved overall survival. And the median overall survival in leptomeningeal cancer is kind of 3 to 4 months. We showed a median overall survival of 9 months. We also showed a clinical benefit rate that was, depending on what you measure, is 75% or greater. And you look at neurologic improvement, you look at reduction of tumor circulating tumor cells, which is our CNSide assay, and you look at MRI, that you're seeing a clinical benefit. And what we found is that our dosing range, therapeutic dosing range, is really on the low end of our original phase 1 dosing. So we've established a recommended single dose phase 2 dose, and we're doing multiple dosing, but we've shown now in the phase, kind of call it 1B, 2A, that we can also deliver multiple doses and get patients to live multiple years. So nobody thought was possible in this group of patients that die within weeks with no treatment and just a few months with treatment. So the signals have been very positive, even early on in phase 1.
So really promising risk-benefit profile in the CNS indications, but just to follow that up, radiation is often used to treat primary or metastases to the liver, and I know that you've got other pipeline assets that sort of do liver-directed therapy. Do you want to just kind of give us an intro into that part of your pipeline?
Yeah, we licensed another drug. It's technically a device. It'll be regulated as a device. It is most similar to 2 drugs on the market that are used for radioembolization for hepatoma. More of a temporizing measure. The problem with those is that they're biomaterials. So you deploy them intravascularly through you put a catheter up into the hepatic artery. You try to cover the tumor with radiation. You block the flow, and you really can't retreat. And so the idea with this drug, we call it BAM. That's its research name. BAM is a combination of 2 different encapsulations: liposome, alginate, and then chelator, and then a different version of rhenium-188 has a better profile for that indication. And we can deploy it essentially anywhere in the body. So another way, get past the blood-brain barrier. If there's an artery you can embolize that covers that vascular supply for that tumor, you can deliver essentially all of your radiation to that tumor. And then over the course of a few weeks, the drug will bioresorb and reestablish patency. So you can go back and retreat. Again, with the idea of turning some of these into chronic diseases. Treat them once. It's like an ongoing way to surgically resect something, but you continue to debulk it from a radio debulking perspective, and then come back and retreat them later. So that's very optimistic about that opportunity. And it's still late stage preclinical, but we think that it has a lot of upside.
Great. So Russ, I want to turn to you now and talk a little bit about CNS side. I know you gave us a little bit of an intro into the diagnostic side of the business, but you want to let's go a little deeper and tell us again specifically. Let's start. What is actually the focus at CNS side?
Yeah, Chris. So we had a test that Plus acquired. I think around a year or so ago, that was already commercial. So, when I saw this opportunity, I've been talking to Mark and the team at Plus since 2024. The fact that it had already been on market, there was demand for it because the standard of care was inadequate. to me was a big positive. They had data on adoption. The test was growing nicely in the hands of the previous owners because it's very sensitive and it's definitive. And what you need to be able to get these patients diagnosed and on treatment is you need some certainty. And we've got a test where we take an LP, lumbar puncture. So we take a CSF, cerebral spinal fluid specimen. We process that in our centralized lab here in Houston, and we can detect cells, you know, very in a case where other technologies don't detect these carcinoma cells. We can get them down to, one or two cells in a whole draw from cerebral spinal fluid, which is, exquisitely sensitive. And you can understand if, you have to be able to find the cells to be able to characterize the tumor, and that's been the issue. So now, after we detect them, capture them, we can run a series of tests to give the physician a really good profile of what that tumor might be sensitive to. And that's the essence of CNSide, is finding those rare events, characterizing those cells because we have the technology to capture them, and providing all that clinically relevant information to the physician so that they can come up with a treatment strategy. And that's really the first episode of, you have to be able to find the cells to be able to characterize them. The thing that I think Mark and the team at Plus happened upon when they were using this tumor cell enumeration technology in their first clinical trial for RayObic, they found that when the patients were treated, the cell count would go down. So it became a proxy for, how effective is the therapy. And I think that privileged position at Plus saw the value in this test beyond just that diagnostic episode to, hey, we've got a really good diagnostic technology here, but we've got something else that is really unique. We can manage the patient. We can give data to the physician to really manage that patient. So if that cell count, when we do the initial diagnostic workup, we're now looking at what's the count doing as we retest the patient on a longitudinal basis. And I think that's where Plus really probably were first to realize that it's not just a diagnostic test, it's a patient management tool. So you can see in stable disease, you won't see that baseline count. So we give the physician a concentration or a burden, tumor cell burden in the CSF. And if the disease is stable, the count stays the same. If it's progressing, the count goes up. And if the treatment strategy is effective, the count typically goes down. So there's a general correlation there. It's like a gauge on how we're doing. So we've noticed with the physicians that we're working with that have adopted the technology under our early access program, it's the monitoring. I mean, sure, they really want a definitive diagnostic answer to be able to go and treat the patient, but it's, they get really excited when they're treating the patient, they see the count drop and they can say, well, we're effective, so let's continue. And it wouldn't be so important except for the fact that there's published cases where, the primary tumor might be breast or lung or melanoma. Once it's infiltrated the CNS, there is some portion of those infiltrations, those metastases that change phenotype, or they actually change the profile of therapeutics that they're susceptible to. So you can imagine, if you're a physician, you can't automatically conclude that what was effective treating the primary is going to also be effective when the tumor is metastasized to the central nervous system. And this is really beyond the blood-brain barrier. This is the fact that it's a different environment in there. pH is different, pressures are different. And there's a documented published paper that shows that in breast cancer, some of these tumors, they flip, it's called. They're biomarkers such that they are not now susceptible. So you really have to characterize that tumor again as when it's metastasized to be able to give the physician the tools to be able to come up with a treatment strategy. And what we're focused on at CNSide is being that partner to the physicians that can give them the tools and can monitor the effectiveness of what they're doing with the patient.
Russ, I just want to follow up, because for those that may not be that familiar with the diagnostic journey, could you just give a very brief overview of, like, what does that golden path look like for CNSide? Who's ordering it? What's the setting of care? where in the treatment, like use a melanoma patient if you want as an example, just because they typically can often have brain mets. Like when is that, how is that workup being performed in the clinical workflow?
Yeah, so obviously there's a lot of different individual cases, but generally speaking, uh, the patients that we're getting the specimens from have had a primary tumor diagnosed. So they're already under treatment, might be for many years, effectively managing that tumor as a chronic disease, taking an acute thing, applying treatment therapies, treatment strategies, and managing it as a chronic disease. Now, what can happen to that patient over time is that they just get cells infiltrating for whatever reason into the CNS. Different environment, different immune profile, blood-brain barrier, so the treatment for the primary is not effective, and you get proliferation in the CNS. So they can show up at the ER with really neurological symptoms, things like dizziness, cognitive issues, headaches, that kind of thing. Like the kinds of symptoms that are really hard to determine definitively straight away what might be the cause. So eventually, once the initial diagnostic workup is done, that physician, the ER physician, might order an MRI or something like that, or do a lumbar puncture, send it down to the path lab, where they'll do cytology on that patient, which is not very sensitive for this particular condition. And so then they start on an odyssey where what's going on, right? So eventually, these patients, as they progress, they may end up being referred to an NCI center, one of the major academic medical centers, and a neuro-oncologist will be assigned. And that's when perhaps there's a suspicion of what's going on here is leptomeningeal metastases. And that physician then is going to use a combination of standard of care, cytology, MRI, and their clinical evaluation. And we can add that sensitivity and definitiveness to that patient. We can come in and say, you know what, we have detected cells in the CSF, and we can give them a baseline. So that's I would say like a typical or a general path to diagnosis. And what can happen is, because of the standard of care is not that sensitive, is that whole period can take quite a lot of time before it becomes obvious with the standard of care that it is LM, leptomeningeal metastases. And what our mission is to get to the neuro-oncologist first. And fortunately, many of them are already familiar with the test and the technology. Many of these neuro-oncologists in the US have already ordered the test before. So, we're in the process of getting that test back to them so they can use it as part of their toolkit, and then expanding really into the regional centers and the medical oncology community, and building awareness there so that we can try to get these patients diagnosed sooner and get them more options. Because it is a devastating complication for a cancer patient that might have been managed on the primary. When this happens, currently, half of these patients usually aren't treated. They usually go into hospice or palliative care, and the other half, the physicians try to do the best they can. So we think there's a great opportunity to insert ourselves there to provide that definitive answer sooner, get the patient treated sooner, and try and impact outcomes.
So Russ, I was a little bit confused because I think you referred to the assay as sort of early access, but now it sounds like you already have a lot of experience in the market. So where are you? What's your recent commercial progress on CNSide?
Yeah, and I apologize for that. I'm sure it is confusing. So the test was previously commercialized. So before Plus acquired it, it was commercialized out of a lab in San Diego, and they did over 11,000 tests. So we've got a lot of data on the use of the test commercially. We're in the process. We relocated it to Houston, to our lab here, and we've been bringing it up again and doing our own validation studies. And when I talk about the early access program, it's this reintroduction of the test into the US market, where we've got 5 sites that we're working with. And I call this kind of like the preliminaries, where you work with the 5 sites, you do the training, the onboarding, you work with them on how to enter the data for the request of the collection of the specimen. So we're at that stage right now where we're reintroducing the test. with our EAP sites, and we've got demand beyond those sites. A lot of those physicians that used it prior are kind of very, very anxious to get it back on the market. And this year, we're going to scale up. We're just wrapping up this month, actually, we're wrapping up our EAP studies. And we will be doing a full commercial launch in 49 of the 50 states in the US this year.
Wow. Okay. Great. I'm glad we had that discussion. It's a lot more clear now. And so a big commercial milestone as we head into the end of the year, Russ. So maybe, Marc, for you, maybe you could take a stab at answering this question first, and then Russ, happy to have you follow on. But as you mentioned, Plus Therapeutics is a unique company. How are you both looking at building an integrated therapy and diagnostics model at the company? How are the two working together in an integrated way?
It's remarkable, I can tell you. And having been on the therapeutic side for my entire career, and Russ, kind of the inverse of that, he can respond. But it's a challenge from a capital markets perspective, because you always get the question, well, the capital markets, they like to be a type. What type of company are you? Are you a diagnostic company?
Right.
Are you a therapeutic company? But I would say that was an issue 6 to 12 months ago when we first started talking about this, and people thought we were completely insane. Now they just think we're kind of the crazy ant. We're not a lot of trouble, but maybe we're still a little crazy. But we have the highest possible conviction that what we are doing is the way to make improvements in survival. Let's talk about the alternative just real quick. I haven't talked about this, but if you go back 40 or 50 years and look at the change in survival in central nervous system cancers, it's flat. So despite a 300% improvement in survival for all other cancers, CNS is largely flat. That corresponds with the duration that MRI has been in the market. So the model that people have been operating under, hey, I've got this new molecule, or I've got this new therapy or a new device, and I'm going to try it for everything. I know it looks like it's promising in rodents for CNS cancers. I'm going to develop that forward till I hit the brick wall, which is usually the pivotal trial. And so what we're doing now is kind of integrating. I mean, we talk about diagnostics, what we find from the diagnostics, and how that relates to what we're finding on the therapeutic side every day. And the data coming out of that is, I think, going to be of monumental importance to the field. So it's the right way to address what is the most intractable oncology problem that's out there, in my view.
Russ, did you want to add anything to that?
Yeah, absolutely. The other perspective, and being in diagnostics pretty much my whole career, we've always believed this is the way that we impact patients by putting together and iterating around the patient. And if you think about diagnostics traditionally, and I've seen it evolve, diagnostics was in the basement, responsible for 70% of the clinical decisions that are made with patients, but only getting 5% of the budget in the hospital. Mm. So, the diagnostic industry bemoaned the fact that they weren't getting the opportunity to add a lot of value. And what's evolved over the last really 2 decades is diagnostics has emerged as therapies have become more effective and targeted. They've become critical to really finding the right profile of patient from a safety and efficacy point of view. It's really hard with targeted therapeutics that are more effective to do that without some kind of a tool to select the patient. And to me, this is a totally natural evolution, but it's remarkably rare. Mm. You don't see these kinds of companies really focusing and corralling the resources around the patient like Marc and the Plus team have done. It makes total sense to me, and I think it's part of that journey of value from diagnostics where, years ago, it was just data coming from a basement given to a physician, and the physician had to do all the work, all the interpretation. How do I use the data? What data do I use? And then there's a little bit more precision in diagnostics, and now I think you're seeing this evolution to where diagnostics is an indispensable tool to manage the patient. And you've seen the value of diagnostics go up in proportion to these steps in value for clinically. So this economic and clinical value has moved up together. And I think when I see what we have here, it's critical mass. It sounds counterintuitive because small company, you're thinking about large markets, oncology, diagnostics. But what we've got is we've got a very tight vertical focus where we can bring the critical mass, we can bring the knowledge. What's really opened my eyes, and this is the first opportunity in my career I've had to work for a company that is really direct to physician and patient. Diagnostics is very much a B2B business. You create these technologies, you sell them to labs. Labs do the testing, and even that is removed from the patient because the physician is the customer of the lab. So if you can imagine us diagnostic manufacturers, we're way back here trying to think of new ways to impact patient care, and we're multiple ways removed from the patient. So the exciting thing for me is this being this close to the patient and physician and being able to iterate at the speed we can at our size and the agility and the thinking that you can bring around, the comprehensiveness of it is super exciting, but I think also has the potential to have the biggest impact.
So as we wrap up the episode today, I thought I would ask you both one final question. If we're looking ahead now, over the next year and a half, what are the one or two make or break questions that you need to answer either scientifically or clinically for RayObic or for CNSide these are the things that what should clinicians and investors and other stakeholders watch most closely as the year unfolds?
Shall I take a crack at that? So, I think the key for us in the therapeutic side is to get the dose right. I think we don't have the classic, escalate till you get a maximum tolerated dose and then treat the patient. Nobody's ever given these kinds of doses. So getting the right dose, getting the right frequency is critical. I'll go on record. There's no question that RayObic works for GBM. I know what I'm saying. I'm completely sober. It works. The question is the delivery. And for leptomeningeal cancer, it's getting the dosing right. And I believe it's going to work well for 125,000 patients in the US that have it. That's what people should look for. Commercially, can Russ Bradley scale? This is a very big market. We're talking about a niche area. It's a very big market in CSF diagnostics. And if we can scale appropriately and partner with the doctor, the way Russ is talking about, this will be profitable very quickly. And that's a real growth opportunity as a standalone, not to mention sort of a driver of value for the whole company.
So Russ, sounds like the pressure's on you, my friend. So what same question over to you with the spotlight on you.
Well, I guess Mike answered for me, but I think that's true. It's true. Essentially, we have demand. So I'm very focused on the call it customer experience, but our physician partners. That's part of the reason why we did this EAP program. We want to make sure we're ready for prime time. But we've got a lot of different tools to bring to bear to help these physicians manage these patients. So the things I'm looking for in 2026 are building out capacity in our lab here in Houston. And what I mean by that is, we're gonna see more testing through here, but we want to add more clinical value. We want to add more markers. So what we want to do is give the physicians not just the tumor cell count and the baseline, but give them information about what therapeutics might be useful. So seeing the scale, building out the capacity, and seeing that test adoption. In addition to that, continuing the work we've done in getting insurance coverage. We've got some really big ones we've announced publicly. We've got United Healthcare and we've got Humana. We're working with some other big national payers to get coverage for the assay. And that to me is providing part of that experience for the physicians and the patients is getting as many covered lives as we can for this test while we bring it up and make sure it's clinically relevant. So I got a lot of KPIs on my dashboard, but if you ask me at the end of the year, how we're going to hold ourselves accountable, it will be showing adoption of this test because we've solved all those issues.
Yeah, this is a fascinating business model. And I know that as I've talked to other leaders in the radiopharmaceutical space, their challenges are also complex, but different than yours in a way that, from what we've discussed today, because for them, a lot of it is having sort of a radioligand kind of imaging step before they can identify. That's their patient selection process to identify patients for appropriate therapy. And you've got your diagnostics, built within the company like this. So it's an interesting model. So, I know we're up on time, and I appreciate you both joining the podcast today. Super interested and be following you closely. Anything else, last comments you want to say to any, potential partners, investors, or life science folks who might be listening today?
I'll just close by, if you look at our website, you'll see the version of the company 2 years ago. So, very soon we'll be doing a major sort of update of that and our messaging and so forth. And it's largely around what we talked about today. So you're getting a sneak peek of what's coming.
Terrific.
Buckle
Thank
up.
you. Yeah, buckle up. That's great.
Yeah.
Hey, it's been a pleasure. I feel like we could have gone on quite a bit longer, but it's super interesting science. The diagnostic side is fascinating as well. But appreciate you both joining the show, and perhaps we should come back in a year or so and see how your progress is going. So thank you again.
Thanks, Chris.
Thanks.
If you enjoyed this conversation, be sure to subscribe to the RX for Biotech podcast to hear directly from the leaders who are shaping the future of personalized medicines. And if you or someone you know is navigating a care journey, be sure to visit therapyforme.ai to search for a specialist near you. And if you need that extra support, be sure to schedule a virtual telehealth appointment with one of our nurse navigators who can help answer your questions. Thank you for listening.